Hello, everyone. Welcome to today's live broadcast, Gene Expression Profiling During Infection. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by NanoString Technologies. NanoString Technologies is a publicly held provider of life science tools for translational research and molecular diagnostics. The company's technology enables a wide variety of basic research, translational medicine, and in vitro diagnostics applications. For more information, please visit www.nanostring.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Wenjie Xu, Ph.D. Wenjie Xu has been with NanoString since early 2016 as a technical service scientist. Prior to joining their team, he earned his Ph.D. and did postdoctoral work at Columbia University studying yeast genetics and infectious fungi. Dr. Xu did apoptosis and drug discovery-based research at SBP Medical Discovery Institute. He was a senior research scientist at Carnegie Mellon University, focused on gene expression profiling, infectious diseases. Dr. Xu has authored or co-authored 30 publications. I will now turn it over to Dr. Wenjie Xu for his presentation. Dr. Xu?
Yeah, thank you, Christina, for the nice introduction. So I will start my seminar. So today I'm going to introduce how the NanoString nCounter technology, the platform, can be used to study infectious disease, in this case, Candida albicans. So Candida albicans is a major human pathogen. It affects more than 400,000 people worldwide per year. And among those patients, there is a very significant death rate. So there's about 40% mortality rate among the people with invasive infection by this pathogen, even with hospitalized therapy. The susceptibility caused by this pathogen is mostly due to a weakened immune system or the presence of an implanted medical device, such as artificial joints, heart valve, and which basically provides a niche or surface for biofilm formation. And the development of new therapeutics for this pathogen is in the utmost importance, as shown in the two little graphs below, that the length of stay in hospital is significantly longer for patients with invasive candidiasis versus the control group is less than 10 days. With the Candida infection, it's almost a month. And the cost also is more than threefold bigger for the people with the Candida infection. And what we know about this pathogen, Candida albicans, is that it's kind of similar to the baker's yeast. It can grow in the yeast form or the hyphae form. And on many surfaces, this pathogen can grow in a form called biofilm. Biofilm is a mixture of the hyphae and some yeast. And this biofilm formation is closely related to infection. And most of our knowledge about Candida albicans is derived from in vitro cultures, like those you grow on agar plate or in test tubes. It's because it's very easy, to get enough cells grown on those under those in vitro conditions and collect enough cell. You can do cell biology, you can isolate RNA, DNA, and do all sorts of tests. However, what we really want to know or needed to know is what does the pathogen do during a real-life infection. This could be mucosal infection, like a thrush, or a more severe invasive infection when the pathogen actually invades internal organs like a kidney, liver, spleen, and then make colonies inside of those organs, which is actually deadly. And so to study the real infection, we generate mouse models where the Candida albicans is injected or given to a mouse from a food, and then that could cause both mucosal and invasive infection. And as you can see, that red organ, that is a kidney from the mouse, and the white spot on that kidney is actually Candida albicans colonies after the invasive infection. And this is what actually I'm going to talk about today, is the invasive infection and how does the pathogen do during that infection. We can learn from its expression profile, and we can also see how the drug treatment under such conditions would affect the cells, from also how the host response is, when the infection happens. So the challenge of doing those study in vivo is is actually illustrated in this figure. So as in any infection, when you try to get the infected tissue out of a patient or a mouse model, and when you isolate RNA, what you found is that the majority of the RNA or the tissue or the cell you get is from the host. Only a tiny portion is from the pathogen. In the case of Candida albicans invasive infection in the kidney specifically, about 99.9% of the RNA you isolate is from the host. Only 0.1% is from pathogen. And why this is a problem is because in many of the existing gene expression profiling platforms, such as microarray or RNA-seq, it's very difficult to handle samples like this. It's a mixed sample with only a tiny portion of the RNA is what you are interested in. This could cause a lot of background noise for microarray, and for RNA-seq, basically you waste a lot of your reads on the host RNA, and also this is pretty demanding for the data analysis. To summarize, the challenges are first in vivo versus in vitro response. We know now that gene expression in vivo often differs significantly from observations in vitro. So you cannot just study, the pathogen in a test tube and assuming that you understand how the pathogen works during an infection. Well, the second thing is that really there's a great need to study a time course treatment and as well as immune response during a real-life infection. We want to understand both sides of the story. How does the host respond to the pathogen? How does the pathogen survive under the host immune surveillance? And also, in a real life infection tissue, it's much feasible to study antibiotic resistance versus efficacy. And another issue from the in vitro study is that there's always a scarcity of tissue samples. If the type of infection is unknown, samples is divided between multiple individual tests, And also, really, more and more, we need some kind of multiplex methodology that can generate lots of data from very little sample. The third point is, as I just talked in the previous slide, that the RNA, pathogen RNA to host RNA is usually around 1 to a few hundred, is to 1 to tens of thousands. So there's really a very big demand for the platform with a good limit of detection, as well as for the specificity as well. And last but not least is the time to answer issue. When the traditional pathogen test identification could take days, if not weeks, but patients cannot wait that long, for the right drug to be administered. And also, there's a need for really expedient data analysis. Many of the regular platforms, the data analysis would require a few weeks by informatics or by statistician. Again, this really would prevent any research or diagnosis from going ahead at the speed that we want. So today I will introduce the NanoString nCounter technology that actually would meet all those challenges. So first of all, the nCounter is really a sensitive platform. It requires less than 100 nanograms of total RNA for an assay with 800 plex quantification. Basically, you can study the expression level of 800 genes, and in many cases, including proteins, in a single assay. And also within a single assay, a same tube, you can study multiple pathogens as well as the host response. And the workflow is really simple. There's no enzyme, there's no library preparation, and there's no amplification needed. And you can even use cell lysis directly if you have multiple samples to handle with, and you don't want to do the RNA isolation for each one of them. And the all hands-on time from RNA to the data is only 15 minutes. That 15 minutes is for one cartridge worth of assay, including 12 samples. And the overall sample to answer time is about 24 hours. That's including your RNA prep, and you load on the machine read out. And so this is really ideal for any time course studies as well as for pathogen identification. And this system really got all bases covered. You can use this platform for identifying what pathogen is causing the infection, and you can do gene expression profiling for the pathogen. And with the same system, same RNA, you can also assess immune response from the host, as well as drug treatment responses. So first, before I get into the real data, I want to give a very short, brief introduction about the nCounter technology. So this is a digital hybridization-based technology to count target molecules actually one by one. So it's purely digital. So the chemistry is illustrated on this slide. We use two probes, we call it half-site. So the capture probe with 50 bases that can bind to the target is linked to a biotin, and the biotin will help basically capture the target and attach it to the imaging surface. And the second half of the probe, called the report probe, would have 6 positions and 4 color barcodes linked to it, so that our imaging system can take a picture of that barcode and know which gene this barcode represents. So with using those two half-sites chemistry, we really can capture the target very efficiently and very specifically. So this slide is showing you one image from our digital analysis system. And for each assay, we capture from a few hundred to a few thousand such images for each sample. So if you enlarge a part of this image, this is exactly what our system can see at the molecular level. Each of those different combination of barcodes is actually attached to a target molecule that which you cannot see. So this is really one-to-one correlation between your target and the barcodes. So each of the barcode that we can count here actually by our system, which is 100% automatic, you can actually read out all the target molecules one by one. And for each assay, the capacity is about 2 million counts. So all the genes that you include in the code set, which is up to 800 different targets, they will share about 2 million counts. So you can, so for gene A, so the next page showing you the output of the data from our system. So as you can see here, for the gene called SPP1, in group A, there's 8,000 counts. So basically, that means we can detect 8,000 copies of that gene in the sample. While in group B, there's only 200 of that transcripts, and in group C, there's 948 copies. And then you can do a simple division between those columns, and you know the expression ratio between the different samples. That's really the basic of what you can get from those samples. We also provide a free software called nSolver, where you can download the data from the digital analyzer and put the data into the nSolver. So the nSolver can perform all the background subtraction, all the normalization for you, and also build the ratio between the samples for you. And more than that, the nSolver also have those some statistical package included, and that can generate a lot of useful plots, such as box plot, scatter plot, and heat maps by clustering both samples and the genes based on their expression levels. So essentially, once you get the data, you can use the nSolver to generate all the useful figures for understanding of the data and for publication as well. So this slide showing you the flexibility for the sample input that nCounter system can take. This is a plot between the purified total RNA, 100 nanograms of that, and then on the other axis, that's the blood lysate. So this is showing you that the cell lysate, direct lysate, and the purified RNA actually have very good correlation in accounts for all the genes being analyzed here. And also the system, you can also use FFP samples, fine core needle aspiration, and samples. It's very flexible. As long as you get enough nucleic acid, RNA, DNA, in some new applications, even the protein, which we label it with some nucleic acid, you can detect with this platform, and it's all in the same lane, same tube. So now I will get into the data from the candidate outcomes in this infection study. And before I start, I want to mention that all the data I described today here is from 2 papers that I published in 2015 and early this year. The first paper is the main study where we performed all the assays on the mouse model, and the second paper in Journal of Visualized Experiment. This is a method paper. So this paper not only contains a very detailed protocol of how to perform the RNA isolation as well as NanoString nCounter assay for infectious disease, for pathogen specific studies. It actually shoots a video of me doing experiments. So if you are interested in doing similar assays, if you want some guidance on how to perform those assays, and if you want a very detailed protocol for your reference, you should search out the second paper, the methods paper. And both papers are actually publicly available. You don't need any access code for that. And the work is funded and performed by Alan Mitchell's lab at Carnegie Mellon University in collaboration with Scott Phillips' lab at UCLA. So now just some experimental setup for the study. So what we do is we use a mouse model. We inoculated the mouse by tail vein with Candida albicans, and then that's called time 0. And then after 12, 24, and 48 hours post-infection, we would sacrifice the mouse and then take the tissue out. The tissue here is actually the kidney, because kidney is the main target organ for a Candida albicans infection. And then we isolate RNA from the infected kidney tissue, and then we perform NanoString gene expression profiling. We use 2 different code sets, a 250 gene environmental response gene set, and a 230 target transcription factor code set. After that, we also started the test for fungi treatment using the same infection model. And lastly, we also quantified host immune response during the same infection timeline. So the first experiment we did, we called it a knowledge-based approach. So as I mentioned before, we have a ton of knowledge about gene expression, about mutations, about what kind of condition is favorable or unfavorable for Candida albicans from in vitro studies, right, from the study on Petri dish test tubes. So as we know that NanoString is not genome-wide, which means we do not study every single gene in a genome. In the case of Candida albicans, that's 6,000 genes. Plus or minus. So we needed to select genes of particular interest for infection or for drug resistance. So how do we come up with which gene to study? So what we did is we collected all the data sets that is available, that is published to date. Those data sets are mostly from microarray, and some recent ones are from RNA-seq. And so those include studies about zinc or iron limitation, oxidative stress, or osmotic stress, and also exposure to serum or 37 degree, which is a kind of a mimic of the in vivo conditions. And also this includes mutations or signaling pathways changes, and as well as for hyphal and biofilm growth, which is directly linked to infection. So I want to stress that all those studies are mostly in vitro studies where RNA is easy to get, but they tell us a lot of things about the Candida albicans. And from those studies, we collected 250 genes that are representing diverse environmental response pathways. And we made probes against those 250 genes, and we use those probes for the in vivo study, which I'm going to present. So here is a figure showing you that what happens to those 250 genes during the infection at 0, 12, 24, and 48 hours. So the baseline is 0 time point, where the Candida is right before it gets injected into the mouse bloodstream. And then at 12, 24, and 48 8-hour time point, we collected samples from 3 to 5 mouse, and then we do the profiling, calculate the average expression level, and made this figure. So what you can see is that there are genes that is upregulated, which is shown in a bright yellow color here, at all time points, starting from 12, and that is gene is up, and all the way to 48 hours. We didn't carry the experiment much longer than 48 hours. That is because the mouse is starting to get really sick, and some die after that time point. So the cutoff is at 48 hours. And there is a gene that is actually downregulated, which is shown in blue, from the beginning to the end during this infection time course. And more interestingly, there's genes that with a dynamic change during that infection timeline. So for example, you can see there's some genes that is blue, which is downregulated at the beginning, and that turned yellow, which is upregulated towards the late part of the infection. You know, I want to stress that actually make a lot of sense, because turn out those genes that is turned on later is actually some of the oxidative stress genes or iron acquisition genes. So we know that neutrophil actually infiltrate into the sites where the infection happens, and that actually poses an oxidative stress to the pathogen, and the pathogen surely responds to such stress. So that all fits pretty well. So now we have a very nice data set for the in vivo infection, and how does that compare to previous data sets, from numerous other labs where they performed a nice experiment on in vitro. So it turns out that the in vivo, which is on here labeled as kidney 12, 24, 48 hours, kind of reproduce some of the signature, some of the gene expression profile that is shown on the a number of conditions, such as exposed to serum 37 degrees, the osmotic or oxidative stress, but none of the in vitro one fully represents the profile that we observed from this in vivo study. So this tells us in vivo is really complex and a unique environment that the pathogen needed to respond to in order to survive and in order to invade, in order to colonize. So that's about the environmental response gene code set. Well, secondly, we decided to take a more on discovery-based approach. So here we have a more open-ended question. So we don't assume we know anything about what each gene do. So we simply ask the question: how about if we study all the cell wall components or the secret protein of a pathogen? Ask which of those proteins genes are actually necessary for the infection. Or how about we study all the protein kinases? Or how about we study all the transcription factors? And here I'm going to present the data for the transcription factors. And there's 230 known or predicted genes in the Canada genome that specifies a transcription factor. We made a probe for each one of those, and we applied this probe set to the same RNA prep that we just described. So this turned out that we have some observations that is similar to what we have with our first code set, the environmental response code set. Here again, we have genes, actually transcription factor that is upregulated early on or downregulated early on, and as well as transcription factor that is dynamic, that is on and off depending on the time point you take a snapshot during infection. So one of the transcription factors that is turned on early is that one, and that is required for zinc acquisition, and this fits very well with what we learned from the environmental response gene code set, where all the zinc acquisition genes is turned on, and as well as all the genes for iron acquisition that is also turned on early on. And an interesting one that is downregulated called NRG1. This is a repressor of hyphae growth, and we know that immediately after the cancer cells enter the host, it will start to form hyphae, which indicates that the pressure of the hyphae growth should be turned down, so that everything fits pretty well. So the next question is that, okay, now we have a data set where a lot of genes, in this case transcription factors, are studied, and we know some of them are upregulated, some of them can kill the mice around day 5 or 7, and those transcription factor mutants actually failed to do so. So, as you know, that we pulled out a lot more transcription factors with the same expression pattern. So, obviously, we can study those transcription factors to see whether they are indeed also play a critical role for infection. So again, you know, this data set serves as a nice hypothesis generator. And lastly, we also used the same RNA and looked at the host immune response during infection. And in parallel to what we observed from the pathogen side of the story, we noticed that there is host immune genes that is turned on early, such as TNF-alpha, IL-6, MINCO, and Dactin-2, and while there is other immune response genes that is turned on at a much later time point at 48 hours. So this includes IL-17, NOX, CCR2. In the figure B here, what I'm showing you is that we compared the overall immune response from 4 representative genes when the mouse is infected either by a mutant strain where some of the key transcription factors has been knocked out, or from a complement strain where the gene has been put back to the mutant strain. So in all 6 cases, there is a significant difference in the amount of immune response that is induced by the complement strain versus the mutant strain. And this is not that surprising because we know that the mutant strain fail to really kill the mice. So the colonization level, the number of CFUs from programmed tissue is much lower from the mutant. And this is also reflected from the more muted response from the host immune system. And we also compared our study, which is a kidney infection, invasive infection, to a mucosal infection, where basically it's a flush that where Candida infects the tongue. And what we observed that there's some genes that is turned on in both type of infection, but there's genes that are turned on only in the kidney infection, and there's gene turned on only in the tongue infection. So this tells us not only that in vitro and in vivo expression profile is very different. In fact, different infection sites, different infection types also would induce very different gene expression profiles. And lastly, an interesting thing we did is look at the response to cathepofungin, which is the main drug used to treat Candida albicans infection in hospital. So what we learned is that there's actually 8 genes that is turned on, induced by the cathepofungin treatment in vitro. By that we mean cells grown in a test tube in a flask with in RPMI at 37 degrees with serum present. And there's about 64 genes that is induced in vivo. However, there is only a small overlap of those genes that is commonly induced in both conditions. There is actually 18 of those that is induced by fungi in both in vitro and in vivo. So this tells us one thing is that the response to the drug treatment, or to actually, in a more broad sense, to any therapy, is quite different in vivo in a real infection versus in vitro in a test tube. So this illustrates the importance of studying the drug response or develop new drugs on the in vivo conditions, where you will get a more true response. So that's the data I'm going to present today. So finally, I want to give a summarization of the system. Why NanoString system would be a feasible platform to perform this kind of study. So first of all, NanoString is very flexible in the kind of input you can give it. It's you can give FFPE samples or lysate, the cell lysate, and also you can give it mixed RNA samples where your pathogen of interest may occupy maybe a very tiny portion of it. Secondly, the workflow is really simple and fast. As I said, hands-on time is only 15 minutes, and the overall time is within 24 hours. This makes it feasible for multiplexed, multiple sample time course studies. And also the data is very reproducible and precise. Each one barcode is a single target, so it's really one-to-one digital counting. And you can assay up to 800 genes at a time. And the data analysis is really straightforward. The end solver basically does it all. So in summary, the high sensitivity, the specificity, and the reproducibility of the nCounter system, really make it a feasible platform for infectious disease research. Another important thing is the system have a great dynamic range. We can accurately quantify genes from less than 100 copies to up to 1 million copies. So there's a 10 to the 5th range of dynamic. So you can assay both high expressors and low expressors in the same assay. And again, the simple workflow and the straightforward data analysis really help to move forward your research in a much faster pace as compared to other platforms. So with that, I can take questions. Okay.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Okay, Dr. Xu, our first question is, can we use this technology for proteomics?
Yes, actually, it's our new application called 3D biology. So what we do is we were able to use antibodies to label the protein of interest with a nucleotide acid tag, and then we can use exactly the same chemistry to detect proteins of interest. Right now, we have two kinds of offering. One is we have a pre-made panel with 30 proteins that is related to mostly immunology and then you can that's off the shelf. You can buy and detect that. Also, we're offering a customer antibody labeling service where you can pick up to 30 of your protein of interest, and we will and also tell us which antibody that works great in your hand for those proteins in Western blot or in other applications, and then we can actually label those antibodies for you, and you can then use those for doing something like a proteomic. Although it's wouldn't be in hundreds or thousands of proteins at this time, but you can easily do 30 proteins, and that you can do the proteins actually together with the mRNA, microRNA, or DNA. So that's why we call 3D biology. So yes, the answer is yes, you can do that.
Okay, next question. In your opinion, has NanoString been more beneficial for discovery or validation in your research, or both, and why?
I would say both. So I think I see that NanoString, while not being actually genome-wide, but it does have the advantage of, as I said, simple workflow and pretty high multiplex. So essentially, as one part of my presentation today, you can actually put up to 800 genes of interest that would easily cover all the cell wall protein or all the transcription factors or all the kinases from any pathogen genome, and put that into one code set, and that would be a great discovery tool or hypothesis generating tool. On the other side, if you already done microarray or an RNA-seq where you narrow down of the gene of interest to a few hundred, then NanoString would become the platform of choice if you want to carry along validation or drug development type of assay where you need to test a large number of samples under numerous conditions. Then NanoString is way more efficient and accurate than the competing platform. So I would say it's really there depends on your needs. We can do both a discovery or validation type of study with this platform.
Next question. Have you ever used other tissues such as spleen, liver, or lymph nodes?
Oh, yeah. Essentially, this platform is tissue, I would say, independent. As long as you can find a protocol that would be enable you to isolate RNA or nucleic acid in that sense from the tissue, then you can use with NanoString. And also, in many cases, you don't really need to even isolate the RNA. All you need to do is to do a lysate. So like a buffer IRT or other kind of lysis buffer, just to make sure the cell break open, the nucleic acid is released. Then you can just apply a few microliters of lysate combined with our code set, and because there's no enzyme, it's directly hybridizing nucleic acid type resin based. So it's really flexible and robust assay.
Now, can the proteomics application be used to differentiate isoforms?
Well, that depends on the antibody. If you have an antibody that can differentiate the isoform, and then our technique can. So the proteomic application is depending on the availability of a good antibody.
Now, what is mostly the biggest difference between microarray and NanoString?
I would say it's digital versus more analog. So in microarray, you look at the fluorescence intensity, between two channels. Here, each of our barcode actually binds one-to-one to a target. So the readout from NanoString is more direct. Each one count of our barcode represents one copy of the transcript. So our data is a lot more actually accurate and reproducible compared to microarray. And also the background level is way lower. We know that our general background is like 10 counts. up to 20 counts. That's it. And our a medium expressed gene with 100 nanogram of total RNA, the readout would be in hundreds of thousands. So compared to that, the background is almost nothing. So we can essentially detect a much smaller amount of RNA and in high complex sample input.
Hey, Dr. Xu, this is a three-part question. I'll read the entire question and we can answer each one as it comes if you like. Can this be used to substitute the actual CFU count to determine the bacterial burden in an infected organ? And is there any validation or direct comparison data? Also, will this be an accepted method by a journal for publication?
Okay, so first, I will answer the last question first. Yes, this is a completely accepted method for publication. Up to now, there's 1,300 papers in all journals, like including Cell, Science, Nature, that used NanoString as its core platform for gene expression, for even for copy number variation, which is a DNA-based assay. The second question is that can this counts be used as substitution of CFU? Yes, I would say yes. Actually, I personally did a comparison of CFU data with the raw counts, of housekeeping genes. Actually, they have a very good correlation. I think that is mentioned in one of my publications, and as well as a number of other recent publications. And also, not only that, actually, I think I saw that at least two recent publications where people developed those NanoString-based assay as a way to detect first which pathogen is involved in an infection from a patient sample, as well as how severe, in other words, the number of CFU in the sample.
Okay. Next question. How much is this technology quantitative?
I would say how much? I would say it's 100% digital. So so this is more or less we promote this as a ratio metric based technology. So essentially our recommended use is let's say if you would have multiple experiment groups, like your control group, your treatment group, or your patient group versus health control group. So the most accurate data you would generate from this platform is when you're talking about the expression difference between two experiment groups. So that's our promoted usage of this system. But I do know that some people try to use also this platform as absolute quantitative method. For that purpose, yes, you can do it, but you need something like a standard curve. So what you do is you can apply a known quantification of the input. For example, since you can easily synthesize known quantitation of oligonucleotide, use that to make a standard curve for the detection, and then based on that standard curve, you can quantify how much of the transcript that you are interested in is from a certain sample. So essentially, yes, we mostly promote the ratiometric usage of this platform. But if you would like to do absolute quantification, you can do it with more carefully mapped out controls.
Okay. Next question. How much is this technology costly compared to microarray?
I do not, it's kind of an apple to orange, because the data you get is not exactly directly comparable. Microarray is genome-wide, as we know, so you can, the plex is in the thousands, while NanoString is more in the hundreds kind of plex. So if you're talking about cost per data point, I would say microarray is slightly less expensive. However, the accuracy is of the data, of basically for usable data point. I would say NanoString would win in that area. And also, for NanoString, because it's high reproducibility, actually we have proven, and this is really accepted by the both the scientific community and the publication, is that technical replicates is not necessary for NanoString. All you need to do is biological replicates. So while in other platforms, microarray or RNA-seq, really you not only need biological replicates, you also need technical replicates. So if you take those kind of factors into consideration, also plus the data analysis, downstream data analysis is extremely easy. Basically, we provide the free software. So if you take all those man hours into consideration, I would say NanoString is really cost-effective platform.
In reference to your previous CFU counts question, can you elaborate on the use for CFU counts for publication, not to the technology itself?
Yeah, sure. So, the CFU is kind of the golden standard that most like labs and hospitals still use that. There's nothing wrong with that. The issue is that sometimes, depending on what pathogen you are studying, sometimes it's hard to culture those pathogens, in a laboratory environment. So the CFU by itself is not also, for example, in some filamentous fungi, the cells are not really easily separable from each other. They make those hyphae and they make a big clump. So the CFU is by itself is not necessarily accurate. So if you compare three things, CFU and histochemistry, where you get some tissue and stain them and count the number of cells there, which is again another golden standard, and also the NanoString quantitation. Actually turned out that the NanoString quantitation correlates with with the histochemistry especially in the case of filamentous fungi, where CFU number is hard to get because of the way that the cell grew, and in a biofilm it's sometimes one CFU not necessarily mean one cell. In that kind of case, there is better correlation even between NanoString and immunohistochemistry. But in general, the more CFU you have pull out from a sample, the more NanoString counts you would get. I don't know if that exactly addresses your question, but that's my comment would be.
We have time for just a few more questions. Next one, how long did it take to analyze your samples with the nCounter assay from sample input to data input for your time course study? Yeah, so I performed this time course study when I was in Carnegie Mellon University in Dr. Alan Mitchell's lab, and in collaboration with Scott Phillips' lab at UCLA. So what we do is Scott Phillips' lab performed all the mouse experiments, and they will just send us the infected tissue, snap frozen tissue to us. So then after I collected all the time course samples, I put them at -80, and when I'm ready to go, it took me 3 weeks to process a few hundred those tissue samples. Basically, I isolate RNA, usually 24 samples in the morning, and then I can perform basically 2 cartridges worth of NanoString assay in the afternoon. So it takes me basically 15 working days to go through more than 200 samples. Then the data analysis take another 1 week or 2. Just plug it into the nSolver and punch out some number and generate some figures. So that, yeah, really from the sample to data analysis is less than a month. Okay, for our final question, are you able to identify multiple pathogens and host responses within one multiplex using the NanoString assay?
Yes. Yes, it is definitely possible and feasible to do, especially for identify multiple pathogens. That's actually a pretty popular application right now. I've seen 2 recent papers where people developed diagnostic assays on the NanoString platform. Essentially, what you do is you design species specific probes. Usually 5, 6 of probes for each species. Just for for the solid, for the basically to confirm your own data, and then you can basically get patient sample, get RNA or lysate, and apply that, and you can easily detect multiple pathogen species. That would be virus, bacteria, or fungi. And the second question is that can we detect pathogen and also host response from the same sample, actually within the same tube? The answer is yes, but that would have one condition. That is the ratio between the pathogen and the host should be within 10-fold. So depends on the tissue type or the infection type. In some cases, the ratio of pathogen cell to host is within a 10-fold difference. In that case, yes, it's very easy to do that. But in some cases where pathogen cell to host cell, like in the case I presented today, is at a 1 to 1,000 level or difference. In that case, because of the dynamic range, it's highly recommended that while you can use the same RNA for the detection, you separate the assay into tubes. The reason is that because the host RNA dominates the RNA, you isolate before you can effectively detect the pathogen. All the reads would be contributed from the host side. So in that case, you still can do the assay from a single RNA isolation, but you would separate your assay into tubes. So that's it. But yeah, the short answer is you can detect multiple pathogens as well as host responses in a single assay.
Dr. Xu, thank you so much for this informative presentation. I would also like to thank our sponsor, NanoString, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through December 8th, 2016. You will receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.
Thank you.